Hello and welcome to this lesson for Advanced Higher Biology, which is still focused on Key Area 2 of Unit 1. And this is the third little section, which is predominantly looking at protein structure. The first lesson here will focus on amino acids themselves and how they are going to influence our protein structure. So prior till now, we have looked at how proteins are synthesised and moved around the cell. We're going to look in a bit more detail about how that protein is actually structured in terms of its amino acids. And then in the next lesson, we'll move on to look at the binding of ligands to proteins and how these can influence their structure. So we are focusing on understanding how the amino acid sequence influences the protein structure. You know this as a fact from National 5 and higher that the order of the amino acids is going to cause the protein to have a specific structure. What we are going to look at now is how that actually happens. So what you're required to be able to do is identify a peptide bond and classify an amino acid. You need to be able to describe the interactions between the R groups of amino acids. You need to be able to describe the primary, secondary, tertiary and quaternary structure of proteins. And finally, explain how changes in pH and temperature can affect the interactions of a protein structure. Now, it's worth remembering just now that proteins are, of course, polymers of our amino acid monomers. There are many different types of amino acid, however there's only about 20 which are used in the synthesis of protein. Now all our amino acids have a central carbon atom which has 4 groups bonded to it. So we have an amine group on one side, a carboxylic group on the other side, and the remaining 2 bonds are taken up with a hydrogen, and finally an R group. And this R group can vary from amino acid to amino acid and comes under 4 main forms. So the classes of amino acids are acidic, basic, polar and hydrophobic, and these classes themselves are determined by the functional group of this R group here, and these properties are particularly important in the structure of the protein. So the R groups of amino acids vary in size, shape, charge, the capacity to bond hydrogen and their overall chemical reactivity. So the wide range of functions carried out by proteins results from the diversity of these R groups here. One of the main classes is an acidic or negatively charged R group. So when the amino acid mixes with any aqueous solutions in the cell, the carboxylic acid group will tend to lose a hydrogen and become negatively charged, and that will influence the other types of R group that it can interact with. We also have a basic or positively charged group which has an NH2 at the top. Now when in the cell, that will gain a hydrogen and become overall positively charged. Again, that's going to influence the types of interactions that it's able to maintain or that will occur within that, around that particular amino acid. Our other class is a polar group. So these ones have an OH, an oxygen and a hydrogen on the end. And they are very slightly charged, giving them the capability of forming hydrogen bonds with other molecules, such as with water. And again, that's going to influence how they can interact, where they're going to be able to be in and around the cell, and will affect how the protein itself can actually fold. Our final class of group is the hydrophobic group. And in this one, these end with a CH3. They carry no overall charge and do not form any hydrogen bonds with water. So that's why they are hydrophobic. They're not capable of forming any further bonds and are referred to as hydrophobic, so will naturally cause the protein where they are to fold against water. So will fold against any aqueous solutions, anything in and around the cell. So they tend to be found on the interior structure of the protein rather than on the exterior. And we're going to look at those types of bonding interactions over the next few slides. First of all though, it's important to have a think about how amino acids are linked together to form that polypeptide during the translation at the ribosome. So we have an enzyme which you don't need to know the name of, causing what's referred to as a condensation reaction. This is where we have the oxygen and hydrogen of one amino acid is going to bind with the hydrogen of the other amino acid, the amine group over here, and that forms water, which is released into the cell, and that forms our peptide bond and forms our amino acid chain, our polypeptide chain, which would be along here. As that continues, we're going to wind up with a long polypeptide chain, and we have eventually got an NH2 group at one end, which is referred to as the N-terminus, and we've got our carboxylic acid group over here, which is referred to as our C-terminus. And obviously the order in which these different amino acids are attached to each other is going to be its primary structure of the protein, and that will have been influenced by our mature mRNA molecule. So if you remember that the primary protein structure is the polypeptide chain of those amino acid monomers. So this is just a diagrammatic example of that previous chemical bond that I've just shown you. So rather than showing you all the chemical symbols that are contained within these amino acids, that's just a pictorial representation of it. So you can see that the specific order of amino acids is going to be different for each protein. And that's been determined by the order of the DNA bases, which have been described into our mRNA, that after transcription obviously you then have alternative RNA splicing, giving our mature mRNA transcript, which is then spliced and translated to form our sequence of amino acids overall. So that's your primary structure. Secondary structure can come in two forms. We can have either the alpha helix or the beta sheet. And these tend to be the result of weak hydrogen bonds forming between the different peptide bonds in our chain. So if we have our amino acid here, and it has a peptide bond around here and another bond around here, there are some hydrogen bonds forming between the amine group here of one amino acid and the carboxyl group here of the another amino acid, and that's causing it to start to form this sort of helix. So it's these weak hydrogen bonds that are starting to form. So remembering that the hydrogen here's got a weak positive charge, down here we've got a weak negative charge, and that's going to help form our hydrogen bonds. So depending on the location of the amino acids in the polypeptide chain, that may cause us to form this particular helix, which is an alpha helix, or it may form the beta sheet. So the alpha helix is your spiral with R groups kind of sticking outwards, whereas the beta sheet has parts of the chain running alongside each other. And that usually forms a folded sheet as well, which is what these lines are attempting to signify, that that's going to kind of fold up. And the R groups tend to be above and below there. Now, when you see these in diagrams, when they are formed, and you may see pictures of these in questions, etc., the alpha helix is usually seen as a coil like this one, and we tend to use arrows to signify the beta-pleated sheet. So you'll see something along those lines indicating that that might be an alpha helix in there, and we've got a beta-pleated sheet coming along here. And so it's possible for proteins to have both of these present at once. And one of the things that you might see when we look at different forms of proteins is the fact that you can have parallel or anti-parallel beta-pleated sheets. All that means is that they're either running with the C-terminuses at the same end and the N-terminuses at the same end, or one is running C-terminus to N-terminus and the one below it is running in the other direction, C-terminus to N-terminus. And again, if you see any pictorial representations of different proteins, you may see them looking like this. So you'd be able to identify the alpha helix shape as that spiral there, whereas down here we've got our beta sheets. And again, on this one, we've got a nice alpha helix spiral in there and some beta sheets along here, so where you can see them overlapping there, so and running anti-parallel to each other. So you may, for example, see a question like this, and you'd be looking to identify the secondary structure present, which would be a beta sheet running anti-parallel. Now, when it comes to our tertiary structure of proteins, this is where we get into the bonding of the different R groups to each other and their presence and how that can influence how the protein is folded or structured. So remember, we have got 3 of our groups which are hydrophilic, so they tend to align to the outside of the structure so that they can bind with water, or just because they're capable of binding with water. They're not repelled by it. On the other hand though, our hydrophobic R group, where they're present, they are going to align themselves to the inside of the structure. Now we also have something referred to as London dispersion forces, and these are weak attractions between the electron clouds of atoms, and hydrogen bonds happen to be a particularly strong example of those. We also have ionic bonds, which are going to form between ionized, acidic and basic groups. So these groups are quite strongly charged and are going to attract each other. And we also may have something called a disulfide bridge, and that might form as a result of covalent bonds between any R groups which also contain a sulfur molecule. So any or all of these types of bonds will influence how the protein itself is actually folded and give it its overall shape. Finally, we have our quaternary structure. And that essentially is where we have a protein that is made up of more than one polypeptide subunit. And that's formed between bonding between their R groups. Now, just to highlight a potential misconception here, it's often the case that you'll see a quaternary structure with 4 polypeptide subunits. Obviously, quaternary means that it's our 4th level of protein structure. However, that does not mean that the protein needs to contain 4 specific subunits. It could contain 2, it could contain 6, but quite frequently it's exemplified in diagrams as having 4. So just bear that in mind. Now that's not the case for all proteins. Some proteins are in fact complete at the tertiary level and don't go on to form any quaternary structures with any other polypeptides. Now, in terms of our protein structure, we may also have prosthetic groups, and they are any non-protein unit which are tightly bound to the protein and are necessary for its function. So, for example, this protein here is the muscle protein myoglobin, and that has a heme group bound to it, and that helps the muscle essentially absorb oxygen, which is obviously going to be quite useful for the function of the protein. Finally, we also need to consider the fact that there are some changes to protein structure. So we all know that proteins are denatured when we increase their temperature. And essentially what's happening is increasing that temperature is going to disrupt those interactions between those different R groups and between the hydrogen bonds between our different amino acids. And as a result, if we're going to disrupt those bonds, that's going to cause the protein to essentially unfold and become denatured. We also have the fact that some proteins are affected by the pH of the solution that they are in. So the pH will change the ionic interactions between charged groups. So if we have a basic and an acidic group, they've formed a strong charge or strong bonds between them, changing the pH is going to influence that, and therefore acidic and basic groups will no longer be attracted to each other. That will overall change the conformation of the protein. However, often that is actually useful for the organism. The example here is the enzyme pepsin. And pepsin is an enzyme which digests protein, but it cannot obviously be released in its active form, or it would start digesting all of the cell membranes around it. As a result, it only needs to be activated when it reaches its final site, and this happens to be when it is in the stomach. So when it comes into contact with the hydrochloric acid in the stomach, it's activated, so it's the interactions between these sections here are influenced, and we lose this section of the polypeptide. So this is our lost bit. It's going to drop off, and that gives us our active enzyme pepsin. So therefore it's able to break down proteins which have been eaten and help to digest those. So from that, you should be able to identify a peptide bond and classify an amino acid into the 4 groups. Describe how the interactions between the R groups of amino acids, describe our 4 levels of structure of proteins, and explain how our changes in pH and temperature can affect the interactions of protein structure.